Okay, so welcome back, everybody. The afternoon session for this 2021 D3 Drug Discovery, Development and Diagnostics. We've had a wonderful morning so far, opened by the WHO and their third iteration of their essential diagnostics list, really trying to engage with countries so they can adopt and adapt that list to form a national EDL for each country. Some great strides forward there, explained by Francis Musi. We've had FIND talk about access barriers for visceral leishmaniasis and the interplay between access and their particular work in the setting of Kenya. We've had an amazing session on schisto modeling to inform new kind of diagnostic approaches and what that actually means in real world settings, i.e. how does it inform MDA, mass drug administration programs in terms of the dosing, all sorts of touch points there. So fantastic use of diagnostics there. We've also then had quite an amazing session with looking at technology, the various technological advancements that are taking place within diagnostics, including CRISPR, including a new alternative to PCR, LAMP, and other kind of minimally invasive techniques in terms of, again, putting access first and letting access dictate the way technology rolls out and shapes itself to the needs of the people. I mean, that brings us very nicely into this particular session. It's a session that's going to look at diagnostic gaps for vector-borne diseases and the challenges of febrile illnesses. That's quite close to our heart here because we also run a worldwide campaign asking the WHO and UN to ratify an official World Dengue Day. Dengue fever, one of the very underreported, especially in Africa, and we felt that might be an idea to open up the entire session to febrile illnesses per se. So I'm very, very delighted. We've got, I'll just run you through the panel. We've got Dr. Prudence Hamad from the Malaria Consortium, one of the world's largest NGOs, very active NGO when it comes to working within, obviously, malaria, but beyond that. Dr. Aubrey Cunnington from the Department of Infectious Disease and this Digital Diagnostics for Africa initiative at Imperial College London, looking at the new front opening up of digital diagnostics. We have Dr. Samuel Duodu from the West African Center for Cell Biology and Infectious Pathogens at the University of Ghana, looking at the use of multiplex panels for the accurate diagnosis of non-malarial febrile illnesses. We've also delighted to be joined by Dr. Mirsini Kaforu from the Imperial College Department of Infectious Diseases, looking at novel biomarkers to distinguish between different febrile illnesses or the causes of those illnesses. And then from the company Mologic, Dr. Joseph Fitchett from the Center of Advanced Rapid Diagnostics, looking at the development of rapid diagnostic tests for arboviruses and viral hemorrhagic fevers. I'm sure dengue will get a mention in there somewhere. So I think without further ado, I'll ask Dr. Prudence Hamad to, I'll give the floor over to Prudence. Prudence, I don't know if you can hear me there or if you can see.
Yes, I can hear you.
Brilliant. If you could bring yourself back onto the screen. Yep, perfect. And I'll round of applause and I'll hand you over. Okay.
Okay. So I'm going to be talking about the diagnostic challenges and gaps that we have when delivering care in developing countries, particularly. So just to revise why we need diagnostics, we need them to get an accurate diagnostic of a disease, which enables a specific and timely treatment to be provided to the patient. It ensures that the treatment is successful because we're only treating the right disease. It reduces the development of drug resistance, and it improves patient and prescriber confidence and satisfaction. We also need diagnostics to do disease surveillance so we can understand the epidemiology of diseases, which enable better public health interventions and improved disease control. We need to look at what are the characteristics that we need in a good diagnostic tool. And we can see that we need accurate results, so they have to have high sensitivity and specificity, even when there's a low prevalence. They need to be set at a level of detection which will identify the actual cause of the disease, but which could pick up asymptomatic or low density infections. We need the results to be rapid, especially when the patient is severely ill. They have to be low cost, both to the health system and the patient. They need to be robust and easily maintained. They need not to be reliant on external power supplies, and they need to be easily trainable for health workers to use them and properly interpret the results. We need to have in place good quality control systems to ensure that the results are accurate, and we need them to be able to be stored and distributed. We need to have a long shelf life, for example, not necessarily to have cold chain, and to not be affected by temperature, humidity, and dust. Who needs these diagnostics? So health workers at all levels of the health delivery systems need diagnostic tools which are adapted to their particular place of work. For example, CHWs need simple to operate, robust, low-cost tools which give rapid results and which can be easily included in diagnosis and treatment algorithms, which they already have, like IMCI or ICCM algorithms. They need to know which cases can be managed safely in the community and which should be referred to a higher level health facility. Health workers at the first level health facilities also need diagnostics that are accurate and that can drive their treatment, their treatments. And in most lower level health facilities, there are no formal labs, but they need to know that they can send a patient 15 kilometers home safely and that they won't die on the way or when they reach home. And hospital health workers in the developing world have access to labs of very many different levels of sophistication, but lab technicians are often in short supply. They're overloaded with work, making the provision of timely results a problem, and they don't often supply a 24-hour service. So this is a picture of a community health worker working in her village, and this is the sort of way she has to work. So we know that most of the healthcare in developing countries is delivered in these kind of settings. So again, they need their tools to be simple, low cost, and give rapid results. And they also need to fit into the diagnostic algorithms that they already use. And they also need to know when to refer patients. So to just put in some of the challenges and gaps we have. So we need to ensure that the diagnosis is a quality diagnosis. So we might need more sensitive diagnostics, especially in countries which are trying to reach pre-elimination and elimination status of various diseases. They should be simple to use, but you might need more sensitive rapid diagnostic tests for malaria, for example, than current brands that can pick up lower density parasitemias. And the quality of the malaria diagnosis is difficult for community health workers who may have vision problems. They may be colorblind. They may be old like me and not able to see the lines on the RDTs. So actually digital reporting tools can be made available at community level that could read the RDTs, for example. And microscopy is being superseded by RDTs in many countries, and that's a problem, and skills in microscopy are being lost. Quality assurance and quality control systems are often very weak, affecting the reliability of tests, especially those most used in the peripheral health systems, such as RDTs. And so when you're thinking about introducing new diagnostic tools, please think about the quality assurance and quality control systems that need to be implemented alongside the tools, and that storage and distribution systems are adequate to preserve the efficacy of the tool. So when a poor community health worker is in her community or his community, there are certain things they need to know about a fever. What's the causal organism? Is it a bacteria or virus for a start, in case they need to prescribe an antibiotic? So they need to know, will the antibiotic work? Is it the correct one to use for that? And are there co-infections that might be causing the fever? If it's a viral infection, what virus is it? Because most of the viruses have very similar presenting symptoms. They also need to know whether they can manage this at home or they need to refer it to a higher center. Another dilemma, particularly around malaria, is when the fever is vivax malaria, can they safely deliver a radical cure? And therefore, they need to know how they can use G6DP deficiency testing, because the testing that's available at the moment is only really useful at the health centre level. Again, should they include other RDTs which are already available for such febrile causes as dengue, leprosy, leishmaniasis, HAT, and SARS-CoV-2 are obviously at the moment? Is the test good enough? Is the sensitivity and specificity adequate to guide the diagnosis? Is it easy to determine the result of the test? And we need to develop new algorithms for interpretation of these multiple test results. And do we need RDTs for other conditions like diarrheal disease or Helminth's diseases? And can we improve decision-making about referral by assessing severity of disease? For example, introducing the use of pulse oximetry or anemia quantification. And for the hospital worker, they are dealing with a much more complex situation, which is the physiological situation of the patient as well as the actual cause of the disease. So the diagnostic tools have to include the causative organism, and whether there are more than one causative organism, but also the physiological status of the patient, which would include full blood count, liver function tests, acid-base balance, etc. And all such tests need to be fitted into the diagnostic algorithms to guide a comprehensive treatment. And at a hospital level, using the right drug is even more important. So diagnostics for evaluation of antibiotic or antimicrobial sensitivity profiles for these kinds of patients are needed to support therapeutic decision-making. So these are just some of the challenges in the diagnosis when diagnostic tools, when they're introduced at different levels of the health system. So that's the end of my presentation.
Thank you very, very much for that, Prudence. Some very interesting points made there, actually, in terms of the dilemma faced by the healthcare workers. It has a lot of resonance, actually, because we interviewed the government of the Punjab in Pakistan about a couple of years ago about their worst ever dengue outbreak. And some of the points came out that interview with it. Even they didn't know what was going on. First thing they were trying to find out was the cause. And in some cases, they had doctors even prescribing antibiotics because they didn't know what was going on. So a lot of resonance there in terms of the real world issues faced and how diagnostics can help to overcome those. So thank you very much for that. I'm sure there'll be a lot of questions coming through from the audience. And from the audience, it's always good to see who's joining us. So Lillian Wapande from the UGHE in Rwanda. We've got from Kenya, Dr. Stephen Gikuru from Egerton University. From Belgium, Stephen de Klerk from ITM. From the Institute Pasteur in Tunisia, Murad Barhoumi. Helena Uliata from the Ministry of Health in Indonesia. Simeon Mogaka from JKUAT in Kenya. We've got quite a spread in terms of countries. From Gabon, yesterday we had the Ministry of Health, and today we've got Dr. Patrice Nezazi from the CIRMF in Gabon. So a very international audience. Thank you very much for the first presentation, which takes us very nicely, I think, to the second presentation of this particular session, looking at the gaps in terms of vector-borne diseases and febrile illnesses. I'd like to pass the floor over to Dr. Aubrey Cunnington from the Department of Infectious Diseases and the Digital Diagnostics for Africa work at Imperial College. And Aubrey, a big thank you from all of us, because I know you put forward a lot of speakers and you were quite instrumental in helping us to shape day two. So thank you very much from Marianne and myself. And I'll hand over the floor to you, Aubrey. Thanks a lot. Okay.
thank you ever so much for inviting me to speak at this really wonderful and inspiring meeting. Actually, I've thoroughly enjoyed the morning as it runs on into the afternoon now. I'm Aubrey Cunnington. I work in paediatric infectious disease at Imperial College, and in the next 10 minutes, I want to try to convince you that recent advances in digital diagnostic technology have created a really exciting opportunity to transform the future diagnostic ecosystem in Africa. And rather than African countries and health systems having to emulate the traditional diagnostic laboratory models that we see in the global north, there is the potential to pioneer new models of healthcare designed around digital diagnostics. I'm presenting this vision on behalf of the Digital Diagnostics for Africa network, and this is a collection of experts from really diverse backgrounds with a common goal to bring together the transformative potential of digital diagnostics to Africa. We have 60 members from 15 organizations in 11 countries and represent academia, governmental organizations, not-for-profit organizations, and the commercial sector. We're supported by the UK's Global Challenge Research Fund, and they are supporting us specifically to focus on Africa, although I hope you'll appreciate lots of the messages might apply elsewhere at the moment as well. And I'm really delighted that actually there are 8 other members of this network presenting today who are going to cover specific details of the potential of digital diagnostics. But I'm going to try and give you a big picture overview in this talk. So what are we talking about when we say a digital diagnostic? So we're talking about something which really bring the capacity of a diagnostic lab to the point of care, to the patient, all contained in a small device. So a small electronic device, which you can put a sample into, which has integrated sample processing, which has a variety of methods for analyte detection, which will often involve a lab-on-chip technology. These analytes are then compiled into an analysis of results, which may be integrated across multiple different analytes. There may be a step of interpretation, which can provide decision support. And then there will be an answer coming out of this device, either directly on the device or through some other user interface. And it would have the ability to transfer data as well. So do we think that digital diagnostics are fit for purpose? Are we reassured? You're probably all aware that there are WHO assured criteria, which were established back in about 2003 to really specify characteristics that new diagnostics should have to make sure that they really could be applied in resource poor environments. And the first of these is affordability. And obviously, when I show you a picture that looks very high tech, a question would be, could this be affordable in an African setting? And we think that it can be, because these devices can incorporate very low cost mass produced electronic components. But I think that will be a topic for discussion later. Sensitivity and specificity are critical. And these devices are sort of showing that they can combine the diagnostic accuracy and precision of a benchtop molecular diagnostic analyzer into a handheld portable device. They need to be user-friendly, and there isn't really much more user-friendly than something where you can put a sample in the top, you wait a little while, and an answer comes out onto the top of the device as well. Rapid and robust is another set of criteria, and you heard earlier in the previous session from Kenny about a malaria lab-on-chip diagnostic, which can give results in about 15 minutes. And the platform in which these are enclosed is a small, pretty robust box which can tolerate temperature fluctuations, being rained on or being dropped. Equipment free means that it shouldn't need any additional equipment outside, and so hopefully a sample can go directly in without requiring pre-preparation, without pipettes, etc. And if it's small and in the way we envisage, then it's deliverable to end users. More recently, a couple of additional criteria have been added to the assured criteria, which bring us into the 21st century properly. So real-time connectivity is very important. And I think that's the way of the future, allowing data transfer and ease of specimen collection. So rather than having to draw venous blood, can we use capillary blood? Can we use saliva samples? And there are precedents for all of these in digital diagnostic platforms to date. What about the diagnostic capabilities that we could cram into a device like this? Well, I think these are really exciting. So we can have sensitive detection of nucleic acids, first of all. And those nucleic acids could enable pathogen detection. They could enable detection of resistance mutations in the pathogen. And that's been talked about earlier in the day for malaria parasites, but it could similarly be for antimicrobial resistance for bacteria. They could detect host genetic polymorphisms. So this could address the problem that Prudence raised in her talk about G6PD deficiency. We could detect the genetic mutations that underlie that, and that would allow you to tailor the treatment of a patient at the same time as you detect the pathogen that they've got. And I think a really exciting development with nucleic acid detection is that the RNA host response can be used as a way of improving on current gold standards of making a diagnosis of infection. And Masini Kaforo is going to talk to you more about that later in this session. There's also proof of principle that you could detect proteins on this sort of lab-on-chip device. That could be antibody or antigen, and small molecules and drugs could be detected in the same way. And we don't have to limit it to diagnosis. It could be that there are biomarkers of severity that we detect as well. These detections in a digital format are fully quantitative, and that can be really important for determining whether a pathogen is causing the disease or may just be carried. Multiplexing allows you to combine different detections of pathogens in a syndromic panel, for example, or pathogen with severity biomarkers, which could guide your earliest steps of management in the community. These tests can be modular, so you might envisage putting in different cartridges into a device, which would allow you sequentially to detect different pathogens based and might be integrated with one another based on the result of the first one. You might put in a different cartridge for the second test. And they could have flexible operating modes. So you might want extremely sensitive detection of a pathogen if you're doing surveillance in order to know if it's in the community at all. For example, if you were considering trying to detect where there's asymptomatic carriage of malaria parasites, you might flip it into a different mode for clinic mode, where you don't want to detect parasites below a certain limit because they're probably not causing malaria as a disease. And the results could be transmitted in a flexible format, perhaps dictated by who's using it. A community healthcare worker might require a different result format to a clinician in a better resourced hospital. And of course, decision support can be incorporated in this. You might not even want to give anyone a result at all. You might just tell them an action to take based on a result. So we envisage a lot being crammed inside this little box. And when you think about then having it connected to an interface, which might just be as simple as a smartphone, you could use it for diagnosis, severity, and decision support. And of course, if you have it connected to a smartphone, then you've got the opportunity for data transfer in all sorts of different directions. So real-time connectivity gives you real-time data. And the data that goes out could be used for personal health records. It could be used for real-time disease surveillance with geolocation. It could be used for something like quality control. And I think a big advantage could be in controlling logistics of supply and demand. This sounds very mundane, but if you use a diagnostic device, then that's one less you've got in your supply cupboard. So then you need to be resupplied with an equivalent number to the number that you're using. If data is being sent out each time a device is used, you know exactly how many you'll need to resupply, and you can start forecasting that. Similarly, if you know you've detected malaria parasites and you're going to treat for malaria, then you know you're going to need to resupply anti-malarial drugs and other medical supplies that might go with that. And you might even link it to funding for healthcare once you've got the real-time data collection. You can also have envisaged data going into the device. So this could be to optimize its performance locally. To use the malaria example again, you might want to set different thresholds depending on the local transmission intensity. You can have personalized interpretation of results. You can have feedback to practitioners who sometimes never know what the final diagnosis was for a patient they've referred on. And you could use it in pragmatic clinical trials. So, for example, the device could be your means through which you have randomization of patients to different treatment arms. So what impact could this have on healthcare and healthcare systems? Well, we could take high quality diagnosis to the point of need. I think that's the really key thing. So this brings it to the accessibility of everyone and heading towards that goal of universal health coverage. It helps us to achieve personalized or precision medicine. This is the right treatment at the right time for the right person. So by using smart combinations of analytes, this could be better for the patient and more cost effective overall for the healthcare system and society. And it allows us to start thinking about data-driven healthcare, where artificial intelligence can be harnessed to contribute to the decisions that are made. So when we compare African healthcare systems and their diagnostic landscapes with highly resourced settings, we often focus on the gaps, such as the lack of diagnostic laboratories and the distance that people have to travel to get a diagnostic test. And we compare that to the ready availability of diagnostics and healthcare in highly resourced settings. But actually, we need to ask the question, do African countries need to try and emulate that diagnostic system in the highly resourced settings? Or could they have the opportunity to build their own route to the future of diagnostics? And digital diagnostics, I think, allow an opportunity to leapfrog over the need for building all the infrastructure that has to accompany having high quality diagnostic labs close to the patient. And we can bring it all closer to the point of need. So those diagnostic gaps become opportunities. And we think there is an opportunity for African healthcare systems to pioneer new models of care around these evolving digital diagnostics. Perhaps doing this ahead of highly resourced countries where the complexity of their diagnostic infrastructure already makes it more difficult to introduce these novel solutions. And this would parallel the rapid digital transformation that is happening already in African economies and capitalize on the value of data, but ultimately enable universal health coverage and healthy lives and well-being for all people at all ages. So thank you for letting me present this. And if you want to learn more about our work, please visit our website.
Aubrey, thank you very, very much. If I had a hat, I'd take it off to you right now, because that was an amazing presentation. Really excited by this concept that the African countries or the LMIC countries or these resource poor settings that we're talking about in field have the chance to reimagine this diagnostics kind of the whole financing of it, the entire sector, if you want to call it that, through this digital approach. I'm sure there's going to be a lot of questions coming through for the Q&A at the end, but that was absolutely fantastic. I think we wrote "The Future's Here," and it's Aubrey Cunnington's presentation. We mean that, by the way. So thank you very, very much for that. I'm sure there'll be a lot of questions coming later. So thanks a lot for that. And I think that takes us very nicely to the next speaker. We're honored to be joined by Dr. Samuel Duodu from the West African Center for Cell Biology of Infectious Pathogens at the University of Ghana, looking at multiplexing in terms of multiplex panels for the accurate diagnosis of non-malarial febrile illnesses. Samuel, you're there already? Excellent.
Yes, I'm there already.
I'll back off and over to you. So round of applause.
Thank you very much for the introduction. Let me try and share. So good afternoon to you all, and So I'm Samudodo, as was introduced, and I'm from the West Africa Center for Cell Biology and Infectious Pathogens at the University of Ghana in Accra, Ghana. So this afternoon, thank you for the opportunity, and I want to share what we have been doing for the past 5 years on trying to improve on the diagnosis of febrile illnesses in Ghana. So the topic for today is Multiplex panel for the accurate diagnosis of non-malarial febrile illnesses. Next slide, please. So as we all know, febrile illness is one of the common reasons why people seek medical care in Africa and also other parts of the world. And the accurate diagnosis of non-malarial febrile illness remains a big challenge in many malaria endemic countries like Ghana, in the sense that the causative agents or what is actually causing febrile illness is not often identified due to the non-specific clinical presentation and also the diagnostic challenges or limitations. We have a significant portion of febrile illness are usually assumed to be malaria and treated as such. And these actually contribute to most of the drug resistance that we see and also poor treatment outcomes. Next slide. Now, so this presentation actually is going to focus on the use of multiplex diagnostic tools for identifying pathogens associated with non-malarial febrile illness in children living in Ghana. Next slide. So this is what I said. We have been working on febrile illness for the past 5 years. We started in 2016, where we're trying to understand the etiology of febrile illness, because we know malaria is reducing. The bed of malaria is reducing, but we still have a lot of cases coming to the hospital. So we're trying to understand what actually caused, what is behind these increase or the maintenance of febrile illness, even though malaria is transmission or the bed of malaria is reducing. So we did a cross-sectional study, which is hospital-based study from two places in Ghana. Kintampo is in the middle belt of Ghana. It's some kind of a semi-urban town with a population of about 40,000, and also in Accra, from a place in Accra, which is a peri-urban area. So these two areas have different ecological settings, and the people living there have different lifestyle. So it was a good base to collect samples from. And also the transmission, the malaria transmission intensity is quite different. Kintampo is a high transmission zone for malaria, with I think infected dose. I mean, the biting, they have about 300 bites per person per year. And Accra is a low transmission for malaria, which has an effective biting rate for mosquito around 50 or less than 50. So we collected samples from children between 1 and 15 years who comes and present with active fever to the hospital. And active fever, we mean that those that come with temperature less or equal to 37 or 38, and the condition occurring less than 7 days. So we collected a lot of samples. We have more than 1,500 clinical samples from these two areas. Next slide, please. So normally, when a child comes with fever to hospital, it comes with a non-specific clinical presentation, either vomiting, diarrhea, abdominal pain, which is a very common syndrome or symptoms of fever. So it's very difficult to know what is actually happening to the child. So the first thing they normally do is to do a malaria test, because we are from a malaria endemic area. They do malaria RDT, also microscopy, to rule out malaria. And if, depending on the signs and symptoms, the clinician can also, diagnose other diseases like, next slide, please. Like requests for other tests like bacteremia, or probably look for urinary tract infections or gastroenteritis. And these are also, they collect blood, stool samples, urine samples, and they culture these samples to look for pathogens. And also, next slide, please. Also, the use of non-malarial RDTs are also common. I know we have RDTs for hepatitis and also HIV and others. Next slide. So from our study, I mean, these figure just shows, I mean, what was clinically diagnosed and what was actually confirmed in the laboratory to look for the pathogens. And if you look at the malaria, I mean, it's not quite impressive, but at least most of the clinical diagnosis were actually confirmed. So they were able to detect parasites in the blood of these patients, of these children. But if you compare to the rest, I mean, the urinary tract infection, bacteremia, the proportion of what was confirmed was very small, very small, very small. Next slide, please. So if you look at the different pathogens that were found in the urinary tract infections, bacteremia, gastroenteritis, it's a whole broad of pathogens. We have E. coli, strep, and all those coagulase negative staphylococcus, and also for the gastroenteritis, we have Giardia, Hemings, and all those were found. So it's quite a broad pathogen. So we can say that some of the kind of fever probably come with so many infections coming from different pathogens. So it's a polymicrobial infection that we are seeing here. Next slide, please. And we also did serological tests to see whether we can detect these pathogens. And surprisingly, we found a lot also in there. We found Salmonella typhi, Brucella, and others. Okay, hepatitis C, HIV, all those. So which still confirm that fever illness is a polymicrobial infection indeed. So if looking at the test, next slide, please. So looking at all these tests that we do, I mean, if we want to do individual, it's going to be very cost ineffective, and that means it's going to involve a lot of resources and also labor. And we are living in a very constrained environment, where resource limited. Our diagnostic infrastructure is not as good. So if you want to do all these individual tests, it's quite going to be difficult. So our study was trying to do a multiplex screening of this pathogen using what we call a Tachman array. So this Tachman array is a PCR-based assay that we detect individual targets, viral and bacterial targets, and also protozoa. So the Tachman array is a rectum-based assay which detects 3 protozoans, malaria, and others. It can detect 7 bacterial agents and also 16 viruses. And you can actually run 6 samples in a single run. So we tried to do. I must say that this was not a very comprehensive study, but I mean we're trying to understand whether we can use these approach. So we only took 166 blood samples for the screening. So what I'm presenting here is a snapshot of what is supposed to be there. And from our analysis, next slide, please. We found a lot of pathogens. We found, of course, Plasmodium was the dominant pathogen that was found, about 37.9%, almost 40%. But we also surprisingly found dengue virus. We also found Salmonella, Rickettsia, Coxella, HIV. Dengue has never been reported. As I think we discussed before, it's highly underreported. So, but with the Tachman array, we were able to fish out dengue for the first time. And that was the first reports, a molecular evidence to show that dengue is actually is being transmitted or in circulation in Ghana. So we reported these to the Ghana Health Service, the ministry in charge of health issues in Ghana, and they took that very seriously. So Rickettsia and Coxella is also not a normal routine. We don't have a normal routine diagnosis for these pathogens, and using this approach, we're able to detect them. These are all zoonotic pathogens. What I want to say is that a large chunk of the samples we cannot detect, we cannot assign any pathogen to it. So 57%, there was no pathogen detection, and these could be because this is a targeted PCR, so we have specific parameters approach targeting these pathogens. So it's some kind of a restricted analysis. I mean, so it could be that there are other pathogens also in the sample that we haven't actually made room for. So because our specific our probes and also the parameter that we use for the Tachman is very restricted to these 26 pathogens that I indicated in the previous slides. Next slide, please. So we see the potential for the Tachman array that is doing something good. But one thing about Tachman array is that it's a bit expensive, and you need a well-trained personnel to handle that. And the Realtime machine is not something that we find in every diagnostic lab in our hospitals. So we're thinking of something very feasible, something that we can actually use without much problem. So there's another study that we do, which revolves around multiplex peptide array. So this is a serological assay that goes to detect also multiple pathogens. So we have a microarray peptide chip developed, and I might say that this was a project that was developed in collaboration with TIBA in Edinburgh. So also a customized microarray peptide chip was designed with the 26 known febrile pathogens that was used in the TACMA array, and these peptides were spotted on the array chip. And we designed both IgM and IgG-based serological screening, so I can know whether what we detect is actively being transmitted or is just due to exposure. So with this peptide array chip, what we're doing is that we are profiling the circulating antibodies in the blood, and in that case, in a blood spot. So that means we need very small volume, which is an advantage in this sense, because for the TACMAN, you need at least 2.5 ml of blood to have a total nucleic extraction. But with these, get a blood spot. So the assay is still being developed. We have made our chips ready with the 26 pathogens, and we are still optimizing the system to detect these organisms in our clinical samples. Next slide, please. So the finding so far from this work is that there's a detection of multiple target bacterial and viral pathogens in a single biological specimen. So it's a polymicrobial infection that we see with these febrile cases, and our assay is able to detect them. And there's also growing evidence for the widespread occurrence of zoonotic and arboviruses in febrile patients, which is something good to know. There's a critical need for differential diagnosis for effective management of childhood, because it's not everything that comes to the hospital is malaria. As we can see here, we have other different pathogens that are involved in the febrile case that we see. So there's a critical need for the differential diagnosis if we want to effectively manage and treat our children. Now, digital molecular diagnostics have a very important role here, and if these can be coupled to that and deployed in remote settings in Ghana, it will be very useful. So Aubrey, we are still, we are working together on this consortium, and I hope we can provide clinical samples for testing when the listening is ready. Next slide, please. So at this point, I just want to acknowledge, I think Aubrey have done that before. We are part of the Digital Diagnostic for Africa Network, and I want to thank Professor Gordon Awandari, who actually leads this febrile study project in Ghana, and also the PhD and postdocs who have been working on this project for the past 5 years. Thank you very much for listening.
Okay, so thank you very, very much for that. Sammy, we're just coming out of the, I'm just getting out of the slideshow in the background, so we should be with you in a couple of seconds while it rejigs itself. Perfect. Brilliant. Thank you very, very much. A wonderful presentation, a new direction of travel in terms of what's possible with the digital diagnostics approaches, and how kind of I was really taken aback by the differences in clinical diagnosis versus lab confirmed. Massive gaps there that need to be addressed. I think this is such an exciting partnership that you've kind of highlighted there. So thank you very much for that. That brings us to the next speaker for this session. We're looking at novel biomarkers to distinguish between the causes of febrile illnesses. As you just heard from Samuel, it's very underreported. There's a widespread feeling that the arboviruses, zoonotic, as well the disease burden is going to be underestimated because of the underreporting. So novel biomarkers to distinguish between the causes of febrile illnesses. A talk by Dr. Mirsini Kafaru from the Department of Infectious Disease, Imperial College London. Mirsini, could I ask you just to get you there? Brilliant. I'll back off. Thanks a lot. Over to you. Thank you. Yeah.
Great, thank you very much. I hope you can see my slides and you can hear me Well, so today I'll talk to you about novel biomarkers and in particular host-based biomarkers. So I'm going to change a little bit gears in comparison to the previous talk, how we can use them to distinguish between causes of febrile illness. And as you said, I'm based at Imperial College London. So in the recent years, we and others have been trying to establish a new paradigm shift. So instead of trying to detect the pathogen that may be lying in sites that are not accessible or in numbers that are very low so they're not detectable, we've been trying to identify patterns in the blood of patients, and we've been looking at genes and the way the genes are expressed. And these patterns are so specific and so unique that can allow us to distinguish between causes of disease and infection. So, for example, you can see in this graph here how the viral response and the bacterial response are unique and different, and can allow us to use the expression of genes as a means of diagnostic use. So this information is readily available in blood immune cells. And how do we quantify gene expression in the blood? So we use some special tubes. In this case, I'm showing an example of a particular set of PAXgene tubes. And what they do is you collect blood from the patients, and these special tubes stop transcription and prevent the RNA from degradation. And what we managed to get is a snapshot, a picture of how the genes in the patient's blood were expressed at the moment in time that the sample was collected. And I'm sure some of you may be familiar with methods of quantifying gene expression, and there are the targeted methods like RT-PCR, but then there are much more newer methods like microarrays or RNA sequencing that allow us to look at thousands of genes of the whole transcriptome simultaneously in a very hypothesis-free approach. And what we do is we get the data, we get the quantification, we get the levels of expression of the different genes, and we try to identify the best biomarkers, the genes that would their expression would differentiate in the best way possible between different disease groups. And we always try to identify those models and use them to predict disease, cause of disease in independent data sets to make sure that our models and our features are generalizable and have the potential to be used as diagnostic tests. So having this pipeline set up, we decided to try and tackle first one of the big problems, and particularly big in the African continent, the problem of diagnosing tuberculosis disease. And I'm sure many of you would be familiar with this issue and how the tests that we have available at the moment, they're not sensitive or specific enough to fully help us diagnose patients with TB. And the problem is even bigger when we talk about children, HIV-positive individuals, and extrapulmonary cases. So almost 10 years ago, we set out a study with 3 different recruitment sites in Africa, in Kenya, Malawi, and South Africa. And we recruited patients into 3 main groups. So we had patients with confirmed TB, latent TB, and then other diseases that mimic TB phenotypically and are part of the differential. And we ran gene expression microarray analysis for all these patients. And we were very intrigued and very happy to see that there is a very big set of genes that were differentially expressed between TB and other diseases. But using variable selection approaches, we tried to identify the smallest set of genes that would discriminate between TB and other diseases and would have the potential to be used as a diagnostic test. And you can see here that from thousands of genes that were differentially expressed, we managed to whittle this down to 51 originally published, and now down to 3 that can form the base of a diagnostic test. And you can see here how well the 3 genes performed. So you can see how the 3 genes separate very nicely the patients with tuberculosis disease from other diseases with a high sensitivity, a bit less specificity, but overall a very good area under the curve. And as always, we wanted to benchmark the performance against the widely used expert MTB leaf, and we had the data from the output of expert from patients in Kenya. And you can see here that although the expert was very highly specific, and this is very well known, our 3-gene signature, the combination of the 3 genes was highly sensitive. So it opens up the field of using these tests in combination potentially. One other clinical question that puzzles the clinical teams worldwide is how can we distinguish bacterial from viral infection? And how can we identify the patients, the children that need, in this case, the children that need antibiotics that need to be treated for bacterial infection, and separate them from those that have viral infection. And here I'm showing some data from an EU-funded study we recently completed, that by using the best diagnostics available in different EU settings and the Gambia, we only managed to identify with confidence the causative pathogen for the minority of patients in our set. But following a similar gene expression analysis pipeline, we were able to identify 2 genes only that by combination, by measuring them in the blood of patients, we were able to identify with very good AUC, very good sensitivity and specificity, patients with bacterial infection that then can be followed up and be treated with antibiotics, and from patients with viral disease. These are just 2 examples, 2 applications of how gene expression measured in the blood can identify disease groups. And it's been shown recently that host RNA signatures have unprecedented diagnostic potential, and there have been many publications out there for different diseases, including dengue, including malaria, TB, as I mentioned before, Kawasaki disease. And in the last 3 minutes of my talk, I'll try to tell you what we're trying to do at the moment, and probably where the field is heading, the gene expression diagnostic field. And I'm sure you've heard from Prudence just before that trying to identify the cause of fever, it's not a dichotomous question. And the way I described the questions before, I posed them as a dichotomous question. What was asking was, is it TB or something else? Is it bacterial or viral? So as part of the Diagnostics for Africa Consortium and other consortia, we're trying to rephrase this question and make it a little bit more realistic, because in clinical management of patients with fever, the question is not dichotomous. You have multiple causes of fever that you have to consider when the patient's in front of you. So this is what we're trying to do exactly with the data and the modeling, and trying to move away from this binary way of phrasing questions to a more multi-class approach, where having a patient in front of us and being able to measure the genes and the way they're expressed in the blood, we can classify them in different groups. So for example, TB, an inflammatory condition, Kawasaki disease, and then viral and bacterial, but also co-infections, and also go down to the level of pathogen. And very briefly, I'll show you some of the very recent data we got. So we used publicly available gene expression data, and the patients were categorized in 6 different groups. So we have definite bacterial, definite viral, inflammatory, Kawasaki disease, TB, and malaria. And we split the data into a training and a test set. And we corrected for batch effects, we fit a model, and we identified 145 genes that were the basis, the classification algorithm which we use to try and classify the patients into the causes of fever. And you can see here on this slide that even for a very detailed level, for the pathogen level, those 145 genes were able to classify most of the patients correctly into the groups we're showing here. And there were, so for all the groups apart from one, we achieved an area under the curve over 90%. Rhinovirus had a little bit less performance, but a lot can be said about finding a positive result for a child with rhinovirus in their nose. And we were also able to classify the patients in a broad level of classification into fewer groups with even better accuracy. So as part of the Digital Diagnostics for Africa Consortium, we're also thinking about the next step, as highlighted by Aubrey before. So we're trying to think on how we can bring those incredible findings to the patient, to the clinic, and to the clinical teams. And we recently published in JAMA Pediatrics the first proof of concept results. So the first proof of concept approach, where we show that the 2-gene signature for bacterial and viral infection can be measured on a chip and can give us exactly the same sensitivity and specificity as the rather complex microarray or RNA-seq processes. And just a couple of examples on how the results could look like as an interface if we were to give those to the clinical teams. So these are real data from the patients. And for example, we have here a patient that had high probability for malaria on the broad level, high probability for malaria on the detailed level, and indeed they had malaria. We have another patient, they had high probability for viral infection on the broad level, high probability for flu on the low level, and they indeed had influenza. And similarly, we can do the same for co-infections, which is very encouraging and a very real-world problem. So we can do this for 2 pathogens, the viral pathogens, or 1 bacterial and viral pathogen. So this patient had pneumococcus, high probability for pneumococcus, high probability for flu, and indeed they had those 2 pathogens detected. So to conclude, I hope I've convinced you that minimal gene expression signatures from whole blood can distinguish one disease from the other. We think that multi-class classification, which follows exactly the clinical thinking process, is much more real world, and it holds great potential in combination with the brilliant technology that our partners bring. We can potentially offer a solution that is accurate, cost effective, and very portable based on the gene expression measurements in the blood. And I'd like to thank everyone who's involved in this endeavor over the past decade, and the Digital Diagnostics Network for Africa. Thank you.
Thank you very, very much, Mircini, for that amazing presentation. I'm sure there's going to be a lot of questions being asked in terms of the advantages, disadvantages of kind of RNA versus protein biomarkers. Very interestingly, how you tripped into the co-infected reality and the potential application in real world to that. I think that's a fantastic road to take. We've heard VL, visceral leishmaniasis with HIV. So many different co-infections exist, and when it comes to febrile diseases, an interesting avenue to explore. I'm sure you'll have a lot of questions coming through in terms of the Q&A, which we'll come to in a bit. But thank you very much for that wonderful presentation. That brings us very nicely to the last of our speakers in this diagnostics gaps for VBD and challenges of febrile illnesses session. Very happy to be joined by Dr. Joseph Fitchett from the company Multiplex, the Center for Advanced Rapid Diagnostics. So delighted to have you here today, Joseph. Excellent. And I'm going to just say thanks a lot for making time for this and hand over the floor to you. All right. Thanks a lot.
Thank you so much. And it's a pleasure to be here with you all today to talk a bit about the development of rapid diagnostic tests for arboviruses and viral hemorrhagic fevers. I will focus the talk today on 2 infections, on yellow fever and dengue, and if there's time we can touch a bit on Ebola as well. And really the objective here is to bring innovative life science to equitable access, which is the ultimate goal of our activities at Multiplex. So just to start really with a busy slide of partners and acknowledgements, and I know I've missed many, many people here, but really to show that this work only happens in partnership and mainly from people who are not supported to be doing this work. And we're incredibly grateful to all of them. And a special call out to our colleagues at the Institut Pasteur in Dakar, in Senegal, and the World Health Collaborating Center for Arboviruses and Viral Hemorrhagic Fevers, who are essential in our delivery of new products for epidemics. And really just to start with this quote from 2016 by Peter Sands and colleagues in the New England Journal. Infectious disease outbreaks that turn into epidemics and potential pandemics can cause massive loss of life and huge economic disruption. And we all know that from how the world has changed with COVID-19 here in the UK and everywhere. And global epidemics are increasing in their frequency, intensity, and severity. We're poorly prepared for epidemics, Gaps are ubiquitous, and that ubiquitous nature of the problem suggests a system-wide change is needed. And we are trying to make some introductions of new systems that can perhaps help address this in the coming years. We need diagnostics that are accurate, affordable, and accessible, but really they're limited in terms of commercially available products. And so we really need to be shifting the target away from commercial return to ensure that these sporadic infections can have the appropriate technologies they need going forward. And of course, the role of diagnostics for epidemics can depend by the phase of outbreak, whether it's prior to the outbreak for surveillance and early detection. Yellow fever is a great example where it can take up to 77 days from index case to laboratory confirmation at the regional centres. And of course, in that time, it's too late. We're in outbreak and epidemic mitigation mode. There's, of course, early in the outbreak to verify cases, particularly in settings where there is a mixed series of infections that can cause similar symptoms, and then late in the outbreak as we map surveillance and anticipated spread, and of course, hopefully post-outbreak. And so there really is an opportunity for change. And here are just some insights from yellow fever rapid diagnostic development, because there doesn't exist many rapid diagnostic tests for yellow fever, as I'll show you. But we really think we can transform the path for equitable access to these kinds of technologies. And that's really through establishing 4 platforms. The first is open research. And what we've done at Multiplex is partner very openly with IP Dakar in Senegal and Fiocruz in Brazil, the 2 regional continental reference laboratories for Africa and South America. The second is product development, where in the UK we would start with the product development, host visiting scientists in particular from Senegal at the moment, but then shift it the other way around, where they host us and train us in their techniques. Thirdly, shared intellectual property. All of our IP, that of IP Dakar, and even the US CDC, although we haven't needed to use their reagents in the end, openly share that intellectual property to make sure that barriers were removed and we could just focus on seeing whether we can develop a test. And lastly, manufacturing facilities. And I put here UK in Europe and Senegal in Africa, because we've been able to create now responsive facilities that can move quickly in the event of an epidemic. So since 2016, we've started deepening our work in epidemics, and we're looking to add more here, but really going to focus on these 3. There's Ebola, there's dengue, and there's yellow fever. And the yellow fever program, initially supported by FIND in Geneva, was really to address a major public health risk, where there is no WHO-endorsed rapid test and no validated rapid antigen test. There is a multiplex assay by SD Biosensor, but reported performance is quite limited, with IgM at 85% sensitivity, specificity 92, and that hasn't been verified widely. And the price per device is close to $10. And so what we're looking to do is de-link manufacture for epidemics, particularly in low-income settings, to be at the cost of production. And in this case, it would be $1 or less than $1.50. And the concept for moving away from IgM, from yellow fever, has really come from dengue. And really where you can see on the left, sensitivity based on PCR naturally drops for the early days of after illness onset. You can see in the second box in figure, NS1 drops in a similar cadence, a bit later perhaps. And then of course IgM increases between days 1 and 5. And then the combination of the 2 gives you a very nice, helpful sensitivity. And so for dengue, of course, NS1 and or IgM positive results are interpreted as dengue infection. So the work we did in Senegal, thanks to the work of Ousmane Faye, Oumar Ndiaye and Amadou Sall, started with an initial sensitivity panel of the test. So 27 PCR positives, 31 IgM positives, and then about 125 well-characterized negative samples. And these results have literally just come into us this week. I've included 2 series of testing from October 2019 and February 2021. And despite some very high CT values suggesting relatively low viral loads, we see a sensitivity of 96%, where we miss 1 of the 27 samples, despite the late presentations to this data set. And we don't see any correlation, particularly between line intensity of the antigen test and the CT value, which is as we would expect. And so we are now, we believe, on the cusp of developing the first rapid antigen test for yellow fever, something that we have struggled with for the last few years. And we believe the model of looking away from commercial return has helped achieve that. Looking at specificity, we see a very good specificity where we only have one discrepancy. And that discrepancy happened to be here in the multi-cross-reacting samples that were IgM positive for yellow fever, dengue, West Nile, and Zika. The flaviviruses being very, very difficult sometimes to characterize. And on PRNT, this sample had equivalent results for dengue and yellow fever. So the hypothesis they're looking at actually was that it was a co-infection between dengue and yellow fever, and may not be a false positive, but just a discrepancy. So really encouraged by these results. And this has been replicated here, the specificity panel in Brazil, where no cross-reactivity to dengue NS1, and then no cross-reactivity, some additional pathogens of importance there. So turning very briefly to the work on dengue that we're looking to advance under the same terms, what we really were guided by was this map. And this is the map from the US CDC that was reviewed and updated this year, I read on the website. And you can see this light blue across much of Africa, and you can see Senegal in this sporadic or uncertain category. There's nothing uncertain about it. Dengue outbreaks happen every year in Senegal at an increasing frequency, and it's been well documented by the Institut Pasteur de Dakar since 2009. So we need to look at this map in much greater detail, and we need a low-cost, effective dengue test that is reliable, that can be used in urban settings where there might be less malaria, actually, and much more dengue than is appreciated. And you can see that Mauritania is no evidence of risk. But again, our test was evaluated in Mauritania by the World Health Organization there with dengue outbreak samples from 2018. So again, there is evidence. We just need to be reviewing it and collating it better. We also need to appreciate that the United States and now Southern Europe are at risk. The first autochthonous transmission happened in France, Spain, and Italy, 2019, and the US, 2020. And where there can be Zika, chikungunya, of course, there can be yellow fever. And with only 120 to 140 million vaccine supply across 4 facilities in Brazil, Russia, France, and Senegal, it's critically important that we mitigate the risks that yellow fever could pose one day, and dengue as well. Comparing the tests that we're developing, we see a lower sample requirement, 40 microliters for the NS1, 10 for the IgM, IgG, can be up to 120 on some of the NS1s, which is inappropriate, we would argue, for children. Turnaround time, we've looked at 10 minutes to keep it as quick as possible. And in terms of performance we've seen, we have seen quite a good performance across 300 samples for NS1, IgM, and also IgG, which I just note isn't reported here. And you can see the huge variability from the LSHTM International Diagnostic Center landscape of performance of the existing kits. And so something that we really need to address in terms of reliability for those presenting with fever. We can also digitize these test results. And so I think another good way of documenting the epidemic of dengue and seeing what we can do about gathering the evidence base in a much more effective way. But really the most important thing for us has been the specificity. As you saw with the yellow fever, flaviviruses cross-react widely. And in Brazil, the current test deployed has a 50% specificity between dengue and Zika. And with co-circulating epidemics in dengue, it's critically important that we do better. And here, looking at the 73 most cross-reactive samples that they have in the Brazilian Federal Laboratory of Fiocruz, it was 100% cross-reacting on the very good test, the PanBio test, but the one that they use as routine. And we saw 12%, 9 cross-reactivities out of that 73. And we think that's in part because we've integrated virus-like particles for the first time into a rapid test. So displaying each of the 4 dengue serovars in a rapid test through using vaccine technology. And it seems to be certainly making some progress here in the Brazilian setting. And so thank you to Anna Bispo and Flavia Levy, whose work this is. And perhaps just to end on this and some optimism that we're seeing here in Senegal, not only do we think these diagnostics can be developed together, but they will be made in Senegal. There's no need to make them in the UK, Europe, Asia anymore. And so here, the world's first manufacturing facility dedicated to epidemics, led by this gentleman here on the right, Dr. Cheikh Tidiane Diagne, with Ibrahima Pai, Ramata Fall, and Umar Ndiaye being the founding 4 members. You can see now there's 40 staff. And from an idea and a plan to a fully fledged manufacturing facility that's independent, I think the future for health security is bright, and we look forward to partnering with them in the future. Thank you.
Thank you very, very much, Joseph, for that wonderful presentation, addressing some of these unmet needs in terms of dengue and Zika, and very encouraging to see the end slide in terms of access development, in terms of manufacturing of that within Senegal. It's one of the white elephants in the room, manufacturing, and I think that's very nicely put forward there by yourself. We know you're very busy with your COVID work as well, so thank you very much for making time for this. What I'd like to do is to just bring everybody back in terms of the panelists, if that's possible. If you could just hit your video feeds again. We've got some questions coming through, and I've just been told that we have until, I believe, 2:05 for this particular session in terms of questions. So that's about 20 minutes worth of questions. I'd like to start because I know the questions are going to come through in terms of quite specific to your approaches, which is completely fine, and we'll definitely ask those. But just to get a kind of a unified question out there, when we talk about diagnosis prudence, you spoke very eloquently in terms of the who is the diagnostic made for, how they're going to use it, that kind of consideration in this. And I think all of that kind of led into a comma for us, that digital diagnostics, comma, it's really about the data, right? Aubrey, you kind of mentioned this somewhat in your presentation as well. And why I mentioned is in relation to the WHO's roadmap guidelines, NTD guidelines, their NTD roadmap to 2030. One of the major shifts in there is this move toward country ownership of interventions or programs. Those country ownerships are really all about releasing or leveraging political will and releasing financial capital in those countries. To do that, they need an evidence base that's based on data. Digital diagnostics, from what we've seen, leads to this new vista where data is suddenly being churned out that could quite easily, it is going to be used in terms of the evidence base. I think we are getting back to evidence-based policy. I think the last few years of political populism and scientific populism are drawing to their natural end, and perhaps it's the age now of evidence-based policy making. So the question is, it's a bit of a long-winded thing, but the question is that in terms of that data, how integrated to, you mentioned clouds, you mentioned various kind of pooling and sharing of data. How far along that process, once the data is generated, are you going to be involved? Are you integrating too? Are you just a cliff edge for the technology, or are you looking at how you're going to get involved in shaping that data, the way it's used at government level? I think I'll go with Aubrey first, if that's okay.
Thank you. I think that's a really, really important question. It's certainly something we've thought about. So within our network, we have a data work package, essentially, who are thinking about these questions, what might be done with the data, even a whole lot of issues around how the data has to be processed to make sure it's safe and secure, that it's legally processed and everything else. So, I mean, there are a whole lot of issues to consider there. There are some very nice principles for digital development, which were developed by FIND with other partners, which have a whole section on data and making sure, for example, you're using open source tools and that you have as much openness of the data as possible, so that essentially it's interoperable between different platforms. And I think that's what we have to aim for. It has to be something that this isn't going to be a single developer who's going to have a monopoly on all of it. That's not what we want. We would like it to be that everyone who develops in this field could integrate their data for a common good. But there is value in that data as well, I should add, and we have to make sure that value is translated into the health economics of this.
Yeah, that's a great answer. Thank you. In terms of translating that data at the community health worker level, I think you alluded to some knowledge gaps there, Prudence, in your presentation, the earlier slides of your presentation. Is this a role for the NGOs that to use the data generated to then forward into training programs for HCPs, or how do you view the Malaria Consortium's position on that?
Well, the question of data is really important, and I was interested to see that in Aubrey's presentation, which we are working on, that data can be used at the Ministry of Health level and at the district level, particularly in stock management, for example, and also in surveillance, because those are really important aspects. And Malaria Consortium has a program in Mozambique, particularly. It's called the Upscale program, and it's provided community health workers with a comprehensive tool where they collect data, and the data is transferred immediately to the district level, to the health facility level, and to the MOH, because it's no use having data around disease patterns, for example, if they're not used. And I think that developing dashboards, particularly at the district and community level, which shows where the disease is concentrated. For example, if you have a yellow fever outbreak, you need to know very quickly that it's there, and you need to get in with your vaccination program. So I think that having devices that can actually transmit the data immediately to the district and to the Ministry of Health, so that it can be used for all the stocking, the mapping of where diseases are occurring. So having a GIS tool on a data collection tool would be very useful as well, particularly from surveillance point of view. And around dengue, for example, one of the problems in Cambodia, for example, is that they only have 6 sentinel sites. So when they have a big dengue outbreak, they have no idea what the actual spread of the disease is, because dengue, about 60 or 80% of cases are low symptom or asymptomatic and don't get into hospitals. So actually, from a surveillance point of view, having a tool that can really pick up dengue cases that are low viral loads, for example, and being able to really know the extent of the infection so that you can take the preventive measures would be a really good idea.
Fantastic. On that point, that's a great answer. Thank you, Prudence. On that kind of preparedness kind of angle in terms of outbreak, preparing for an outbreak, Joseph, you mentioned your work in dengue there, obviously, in terms of the outputs coming out of your approach. Have you thought, or have you looked at sectors outside of outbreak prevention? I'm talking about vaccination, simply because the current vaccination landscape as it stands for dengue, the serostatus of the patient is a completely vital component of the approach to vaccinating the population. And so are there any moves from Mologic to perhaps potentially partner with vaccine companies looking to get a better handle on the serostatus of the patients?
We haven't partnered with a vaccine company, but we are validating the serostatus in Laos with the unit in Vietnam there. So exactly. So we should have that data in a Southeast Asian setting. We started our validations in Africa. So we have 3 sites from Senegal, Mauritania, and Kenya that validated the kit. We then went to Brazil, and now we are in Southeast Asia looking to see if across the 3 continents in those different settings we see equivalent performance. And so far we do. And of course, serostatus will be very important, particularly in Southeast Asia.
Yeah, fantastic. So I'm going to come back to both of you in terms of how this is going to impact the financing of diagnostics moving forward and the new kind of horizon that's opening up. But just to come to the other speakers as well, we've had some questions come through, quite specific questions. So I'm just going to bring those up. Just bear with me for 1 second. So for Samuel, there's been a question from Murad Barhoumi from the, I believe, the Institute Pasteur in Morocco, asking, can we use the same strategy, multiplex, for leishmania? You'd have to unmute yourself, Samuel, sorry, yeah. I always do that, don't worry. Yeah.
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 So the multiplex system works for all pathogens. I mean, the most important thing, for example, the TACman, if you have a specific pathogen-specific parameters and probes, it's possible to detect any pathogen. So there's no restriction. And also the microarray, the peptide microarray can also be designed for specific pathogens. So I mean, it's not disease restricted. It can be used for all kinds of pathogen diseases.
Okay, fantastic. Great answer. Thank you. No, Murad's happy with that answer as well. Thank you, Samuel, for that. To Mercini from Núria Balançana from Brazil. In your opinion, what are the advantages and disadvantages of using host gene expression profiling instead of host proteins? So I suppose it's the disadvantage of RNA versus protein biomarkers kind of question. Yeah.
Yeah, thank you. It's something we've been mulling over for years as well, and I didn't have time to discuss proteins today, but similarly, the way we're using RNA to try and come up with disease diagnosis, tests and kits and algorithms, we have a similar stream of work for proteins. There are definitely advantages and disadvantages on both. So for example, we know RNA is not as stable as protein, and the technology for protein detection is a little bit ahead of time. So there are dipstick tests like the pregnancy style tests that you can measure protein much more easily at the point of care. But it's scientifically a very interesting question as well. And we've seen when we compare bacterial and viral infections, for example, that the RNA and the protein measurements in the blood, they seem to be capturing different parts of the host immune response. So we see, for example, the interferon pathways being much better captured by the RNA, while the bacterial host response is much better captured by protein. So we are also working towards combining and contrasting the both of them, potentially in a point-of-care diagnostic test as well.
Fantastic answer, thank you. And Núria is also asking, what do you think the role of prognostic severity biomarkers versus diagnostic biomarkers is in febrile illnesses? So prognostic severity biomarkers versus diagnostic biomarkers.
Yeah, so obviously both are extremely needed and useful, and depends on the clinical question for the particular disease. We've seen we're doing some work, apart from the diagnostic biomarkers, we're doing some work on severity progression, and we've managed to identify a handful of genes that seem to be not disease specific, but they are able to predict severity further down the line. So I don't think the same biomarkers can serve both purposes. Okay. You need to do different discovery and different validation for prognostic and severity and diagnostic biomarkers. But it's something that the field needs, and we are actively working towards that as well.
Okay, brilliant. So Professor Dolores Parra has made a point that, well, she's asking, do you feel that the production of tests, and I suppose this is, I'm going to ask this to Joseph because of the work in Senegal and also to Samuel. Do you feel the production of tests do you feel that the production of tests in a no loss, no gain scheme is getting more feasible which are the factors pushing in this direction? I mean, I'd qualify that by saying that COVID has really shone a light in terms of how important this sector is, and how closely involved governments need to be in this sector. But she's asking, do you feel that the production of tests in a no loss, no gain scheme is getting more feasible? Which are the factors pushing in this direction? I'll ask Joseph first.
Absolutely. So Senegal have the first facility to do this for epidemics, and it's the factors pushing it is frustration. It's been decades of no yellow fever test, no reliable Ebola point of care test, dengue in Africa, and then they also are working on measles and rubella. Then COVID came along, so that facility needed to be boosted, which is happening. And then in the UK, we established a social enterprise, Global Access Diagnostics. So even in the UK, we're making tests with no private profit. And you can make these tests in the UK for £1 to £2. And so there's no reason we cannot be doing this in general for epidemics, because the public health impact is way more important than the commercial business plan. And prolonging, profiteering will only prolong a pandemic. And so I hope that this production of tests will be the norm for sporadic outbreak-prone infections going forward.
It's a really interesting model you're putting forward there, the Senegalese model, if you want to call it that, actually, Joseph. So hats off to you there. I think that does answer that question quite nicely. In terms of the financial incentivization of diagnostics per se, let's explore the impact of this the data generated at digital level, how that's going to kind of change the governments that are involved. We're talking about political will. We're talking about investment capital being unlocked in countries. As soon as you say the word digital, it kind of opens up kind of a landscape of new skills, perhaps more centered with the programmers and the kind of financial incentivization. We've got, for the drugs, we've got a priority review voucher. For diagnostics, it's not so streamlined at all. Regulatory harmonization is a big issue in terms of countries and future development. How do you think the digital approach is going to alter that? I'd like to ask, it's a big question to ask, but I'd like to ask Aubrey that first, please. If that's a natural person to ask, it'd be Aubrey. Yeah.
I think I'm going to have to be honest with you that we don't have all the answers to that at the moment. This is going to have to be worked out with all of the potential stakeholders, because it's a totally different model, isn't it? Yeah. Your whole sort of value proposition is around data, rather than just being able to make a diagnosis, because that's what's going to tip the balance in terms of whether these are cost effective or not. Yeah. And you can imagine that the value of data is immense. I mean, that's really how, for example, Amazon has made its huge profits. It's the data, not the selling of the products so much. And I think we need to engage with all of the stakeholders to do this. I'm a clinician. I'm not an economist.
Yeah, I was going to ask you, let's redefine the stakeholders, because I think we're going to have to redefine the stakeholders. Samuel, who should be involved? Who should be sat at this table in terms of having a discussion? We've said data is the new currency when it comes to this approach. The massive impact it could potentially have at government level, new evidence-based policies could be sprung because of this data. So many different kind of avenues to explore. Who should be at the table? How do you redefine the stakeholder for this approach in diagnostics?
Yeah, so as Aubrey was saying, it's very important to have all the stakeholders involved, and the findings that we're getting from the work we have done is very relevant. I mean, in terms of shaping the health policy in the country. And I think what after our first discovery of active transmission, or maybe that dengue is circulating in the country, we actually seek to get more funding from the government to actually expand the work. Okay, because I mean they really appreciated the work that we did, and which obviously needed more resources, to have a good coverage. And this kind of engagement that we're talking about, because investing in such research is very critical for the government, because we need to know what is in there, I mean, what is with us? So at least we can give a better healthcare. So I mean, spending money like what is happening right now in the world with the COVID situation, I think, I mean, we have be able to squeeze some money to actually control the situation. So it's the same way other diseases are very important, because we have a huge infectious disease burden in Africa, and it's about time that the government spend much money in trying to control this situation.
Yeah.
So, yeah.
One of the first press releases from the African CDC when they went public, just a year or so ago, was the need to increase data sharing across the different countries in Africa. And I think so it is, obviously it's vital for everybody to get a hold of that. And I wonder how this will result in a different type of stakeholder moving forward. Fascinating session, actually, to be honest. And I don't think we've got enough time to go through. I can see some other questions, quite technical ones coming through. Paul Hose asking, Mirsini, have you looked at differentiating fungal infections from viral or bacterial infections? All of these things, I'm sure you'll be able to exchange emails. If not, we can send an intro email. Prudence, we wanted to also open up a bit more in terms of whose responsibility is it? Who's going to fill that void in terms of this knowledge gap that you kind of alluded to in your earlier slides? But I think we might have to have a different session on all of these issues. We just wanted to say thank you very, very much. I'm being given the stare that the time is coming to an end. And if it's okay with everyone, if there are any questions unanswered, which I do apologize for if that's the case, you would be all right if we send you an introduction email to perhaps continue the conversation with the people asking the question. Are you all okay with that? Okay, yeah. Brilliant. And then you've got a smile as well, so that's brilliant. Thank you very, very much for a wonderful session. I think we're probably going to have future sessions that open up these approaches in a more depth, and I think we're very happy to do that through our ISNTD Connect sessions. So many questions unanswered. Thank you very, very much for your time. Thank you very, very much indeed. All right, thanks a lot, everybody. Okay, take care. Thanks.
Thank you.
Bye.
Bye.
Thank you.
Bye.
Right.